This is the second video looking at gymnosperms and their naked seeds. In this one, we're going to look at a xerophytic leaf cross-section and review some of the characteristics of xerophytic leaves, and then we will look at the cycads and the ginkgos. So this is normally the point where I would source answers from you and see what those features of xerophytic leaves were, but since I am just talking to myself here, I'll write some down. But you can be thinking, before I write them down, what were those features? So they would have had high volume and low surface area, because surface area is area that you can be evaporating water from, right? So the less surface area we have and the more volume we have, the more effectively we can store water and keep it from evaporating. We also had sunken stomata, and in extreme cases, we had stomatal crypts. We had a thick cuticle, that waxy layer, that's going to help us keep water in. Also provides a little bit of sun protection. Stomata would be always on bottom or hidden in some way. That kind of goes with our sunken stomata. In some of our leaves, we had boliform cells that allowed the leaf to roll up in the case of dehydration. Rolling up would be another way that you could potentially hide your stomata. We also had in some of our xerophytic leaves a hypodermis, which was effectively more layers of epidermis, so a much thicker skin underneath that thicker cuticle. More protection from the external environment and from drying out. Maybe you can think of some more, but I'm going to move on to the next slide. Because we're talking about gymnosperms, the xerophytic leaf that we'll draw will be a pine needle. So you've seen this before in Lab 7, but we'll draw it again just to kind of reinforce those ideas. Okay, so this outer layer here is our thick cuticle. And remember that that's waxy, right? So that is going to keep water from escaping our tissues because that wax is hydrophobic. And then I'm going to skip to another part on the inside. We have this layer of larger cells surrounding our vascular tissue. And much like in the roots, this is an endodermis, an inner skin. So not only do we have a thicker outer skin, but now we have this extra layer within the leaf that prevents water from escaping our vascular tissue. So because we are thinking about this leaf in its correct orientation, the xylem is going to form on top. Of these little vascular bundles. And the phloem would form below that. So in our xylem, because we are kind of pre-angiosperms, we have tracheids, no vessel elements yet. And then in our phloem, which, well, that's a crazy H, we would just have sieve cells, no sieve tube elements. No companion cells yet either. All of the rest of this cellular material in here is called transfusion tissue. And that helps insulate the vascular bundles. Something that's not part of a xerophytic leaf, but that you will see in these pine needles, are big holes. And this has nothing to do with prevention from drying out. This has everything to do with other conditions that were present on the earth when gymnosperms evolved, namely insects. Right, so these are for prevention from herbivory and to quickly heal wounds and keep them from being infected. So these are resin canals. So sticky fluid would be, or yeah, it's a fluid. Sticky fluid would be running through these canals, and it would contain highly toxic materials so that if an insect did bite into this leaf, it might be poisoned by those terpenes and terpenoids, or its mouth parts would just get gummed up. And then that resin would clog the hole or wound that that insect created. So we have resin canals, and those are situated within all of these sort of puzzle piece shaped cells with a lot of surface area to interact with each other. Those are the mesophyll cells. They kind of make up that body of the leaf and provide more volume. If we're looking toward the epidermis, we have our outermost layer of cells, right, our epidermis. And I'm just drawing kind of pieces of this leaf everywhere, so it's not one whole picture. Sorry about that. And remember that epidermis is always just one layer thick. Just underneath the epidermis would be a few layers thick of cells, all small and tightly packed together. And this is the hypodermis. So hypo meaning under, dermis meaning skin. So this is the layer of skin just under the epidermis, which means on top of. Many types of dermis here in our xerophytic leaf. Within the hypodermis is where you would find your guard cells for your sunken stomata. So here in light blue, are our guard cells. Pardon me, sunken stoma. So our stomata are kind of buried within that leaf, and that provides this region right here of air that is outside of this boundary layer. So we would have relatively dry, arid air blowing past our thick cuticle and our epidermis. And if we had direct contact between that dry air out here and our internal humid, wet environment, then water would escape much more easily because it's just diffusing from an area of high water to low water. If we keep this humid pocket that's going to have relatively more water than that external air, that allows for water to move outward more slowly, so we can retain more water inside of our leaf. Here are some images of pine needles. One is an actual image from a slide and the other is a cartoon image, but they give you all of those different components of those xerophytic leaves. let's look at gymnosperm diversity. Our first group that we'll look at are the cycads. I think these are no longer called Cycadophyta, it would be the Cycadopsida, which is, I think, a subclass. So these are fern-like in appearance, except for if you were to feel those leaves, they would be sharp, tough, and leathery, maybe even plasticky, because they are xerophytic leaves. Unlike our ferns, which when I try to bring in samples of ferns to lab, they dry out almost instantly, even if I do put them in water. They're not adapted to these dry environments. Some ferns are, but most of the ferns that we find around our area, because we live in such a humid environment, are not. So fern-like in appearance, but the other thing that they have that's different are these large cone structures, these strobili. So here is a female cone and here is a male cone. That'll look different depending on the type of cycad, but this is where their pollen would be produced here in the pollen strobilus. So it's a microstrobilus, even though it's huge, and then this is where seeds would be produced inside the megastrobilus. And these would occur on different plants. So dioecious might be a new term for you. Dioecious means two houses. That means that the male and female reproductive parts are stored on different houses, different plants. So we have female plants and male plants. Female plants have the megastrobilus, male plants have the microstrobilus. Pollen needs to make its way from a male plant to a female plant. And it does that by insect pollination. And so there's a variety of different ways that we'll see pollination happen. I linked to a In Defense of Plants episode, or I think it's maybe just a blog that talks about different ways that cycads get pollinated and how they use insects. Most of our gymnosperms will be wind pollinated. You can see here we have these cool flagellated sperm. And so that is really similar to our ferns and our bryophytes, where the sperm still have flagella to swim, but instead of only having two flagella, we have many flagella. So very interesting. Now we move on to our ginkgos. So we only have one species of ginkgo in the entire Ginkgopsida, which I believe is correct. And it's almost unchanged from the fossil specimens that we have. So this is a living fossil and the only remaining member of its entire, used to be phylum, now class or subclass. So these are trees that can be quite large. They're deciduous, so unlike many of our gymnosperms who will keep their leaves year-round because they're sort of high investment, these xerophytic leaves, ginkgos will lose their leaves seasonally each year. So they're deciduous, and they have these really distinctive fan-shaped leaves. And this is exactly what we find in the fossil record, are leaves that look just like this. And here is a huge tree with the leaves falling off. They turn this beautiful golden color. Ginkgos also are amazing in their ability to live thousands of years. I linked to something in the lab. It's an article about the sort of immortality of ginkgos and the things that trigger their senescence and sort of how those are lacking in ginkgos. Ginkgos are also dioecious, so they contain female parts on one plant and male parts on another plant. And interestingly, they have structures that look a lot like angiosperms. So if you look at these male cones, the microstrobili, actually look a lot like male catkins that we'll see in angiosperms. So keep this image in mind. They look just like anthers in a catkin. And then our female cones are these paired ovules that sit at the ends of these branches, and the seed is inside here and is actually inside this kind of fruit-like coating. So that is very similar to what we'll see in angiosperms. Angiosperm means seed with a vessel, right? And so that's sort of what we're seeing there. They are wind pollinated, but they have the same kind of appearance of flagellated sperm with many flagella as the cycads. So very weird. I mean, it doesn't really matter. The pollen have to get delivered and then they produce the sperm, and then the sperm have to swim through a tube to get to the archegonium. So it makes sense that they're flagellated, but just strange how they look so similar and so different from everything else.